Hey, and welcome back to our second of the four macromolecules. Today, we'll be discussing proteins. So starting off with the structure of a protein, we see the image here. Proteins are made up of monomer units called amino acids. So here is a general picture of an amino acid. All amino acids have three main parts. They have an amino group. Amino is one N and two H's, so here that is in blue. They have a carboxyl group, which is a carbon double bonded to an oxygen, and then single bonded to an OH, a hydroxyl group there. So that's a carboxyl group. And then finally, all amino acids have a side chain, which is symbolized as R here. We use R kind of like a variable in math class. R can be a lot of different types of compounds. There are 20 different side chains, so that means there's 20 different amino acids. They all have an amino group, they all have a carboxyl group. What's different about them is their R group. So again, there's 20 total, but here are 4 of them, just to give you an example. So you'll notice all 4 of these amino acids, here they have their amino group, here they have their carboxyl group, there's the carbon in the middle, there's the hydrogen that, in the image up above, the hydrogen's on top, but in this image the hydrogen's off to the right. And then the R group. Glycine, the most simple of all amino acids, just has a very simple H. Just a hydrogen, that's the whole entire side group. Alanine, another very simple amino acid. Again, it has the amino group, there has the carboxyl group, there's the hydrogen, and its R group is just a methyl, which is carbon and 3 hydrogens. Pretty simple side group there. Proline gets a little bit more complex. You can see proline has a little bit bigger, a little bit different shape. And here's valine. Again, a little bit different shape. And that's what makes each amino acid unique, is that side chain. Some are big, some are small, some are circular, some are shaped like a Y. All sorts of different sizes, all sorts of different shapes. And we won't get much into this in this class, but all sorts of different reactivities as well. Some react with acid, some react with temperature. So kind of continuing on from that last page. When you change the acidity that a protein is in, when you change the temperature that a protein is in, you can change the way that those R groups interact with one another and interact with their surroundings. When that happens, you can change the shape. So a protein has a very specific shape. If you remember back to the very first day, when we talked very briefly about proteins, we mentioned that they have like a lock and key type model. That key has to fit the lock. Shape is very, very important for proteins. And that shape is determined by the sequence of amino acids, whether that R group is big, whether it's small, whether it reacts with water, whether it reacts with acid, whether it reacts in hot temperatures, low temperatures. The way that side group, that R, whatever it is, the way that it's shaped is going to help the protein make its proper shape. So hey, here's a substrate. Proteins work on substrates. Hopefully we recognize, based on the hexagon shape, what substrate we're looking at here. This particular protein, it looks like it's going to be fitting these hexagons perfectly. More than likely, this protein is helping the digestion of carbohydrates. That's what it looks like this protein's job is. See the exact correct shape here, and that's good. That's a well-shaped protein. If that protein is subjected to a different pH, different acidity, or a different temperature than what it's used to, those R groups might start to react differently. The whole protein may fold differently, and boom, not good news. We have what we call a denatured protein. It's when a protein has the wrong shape. So you'll notice it no longer has that hexagonal shape. It's not going to fit this substrate the way it should. This protein probably can't do its job because it is denatured. So acidity and temperature are both very, very important when it comes to protein folding and proteins doing their jobs. Right, on to the formation of proteins. This should look really, really familiar here. Amino acids, again, there's the monomer unit of proteins. They get linked together through, hey, guess what? A dehydration synthesis. There it is again, exact same as the carbohydrates yesterday. The hydrogen of an amino group is going to bond with the OH, the hydroxide of a carboxyl group, and boom, H2O leaves. Dehydration, water leaves, synthesis. The nitrogen will now be bonded with the carbon, and we call that bond, very special type of term for this bond, a peptide bond. So it's a peptide that links two amino acids together. So here we have an amino acid, there we have an amino acid here. They're linked together. We have a growing polypeptide chain. A lot of proteins are made up of hundreds, sometimes even thousands of amino acids. Each of them have a peptide bond between them. So a common nickname for proteins, as you're forming these proteins, is a polypeptide, many peptide bonds between them. So proteins are made, polypeptides are made at what we call ribosomes. Our cells have two types of ribosomes. We have free ribosomes, which are kind of just floating around all by themselves anywhere in the cell. And then a lot of times we have what we call bound ribosomes, which are attached to what we call the rough endoplasmic reticulum. We'll cover this more in chapter 4, I believe. We'll talk more about ribosomes, but for now, we just need to know that proteins are made by ribosomes, and ribosomes can be free, floating around the cell, or bound to what we call the rough endoplasmic reticulum. We can make our proteins, and we know what shape they should have based on their amino acid sequence, and that amino acid sequence is determined by the genetic code. And that's how we get from DNA, our genetic code, to, well, all the things that make you you. The shape of the proteins, the type of the proteins, the way the proteins behave, is all determined by the genetic code. And that process going from DNA to protein is called translation. We talk about that in Unit 7. All right, the function of proteins. Well, proteins are the workers of the cell. They do everything from support to storage. They transport materials. They signal one another. They are involved in the movement of cells, the defense against foreign substances. Oh man, you name it. The work of the cell is done by proteins. There's a very special type of protein. We'll talk about this more in a later unit as well. But there's a special type of protein called enzymes. And the job of enzymes is to accelerate the chemical reactions in your cells. All the reactions that are occurring in your cells, again, that's called metabolism, they would not occur without enzymes. Enzymes speed those processes up by thousands to tens of thousands of times faster. So again, your cells wouldn't exist. They would not be able to perform all of those reactions that they need for life without enzymes helping them speed those processes up. So kind of an example of how important and how gigantic proteins are in a cell. If you take a human cell and you find out the mass of the human cell, most of that mass is going to be water. Water makes up a lot of our cells. But if you dry the water out, all right, remove the water, find a dry cell, about 50% of the mass is going to be proteins. So you can keep in mind, we've got 4 types of macromolecules. Proteins, and for humans at least, represent about 50%, even though they're only 1/4 the total number of macromolecules. They're about 50% of the overall mass. So just kind of a heads up as to how big and important proteins are. Hey, one more time, because this is very important and you're going to hear it a lot, the specific shape of the protein is very, very important for how the protein works. So the example I have up here is an antibody, which is a specific type of protein, and it's going to identify antigens on the surface of a foreign bacteria or a foreign cell, whatever that foreign cell is. So the shape of that antibody is really, really important. You'll notice the antigens, which are the surface proteins on the bacteria, have kind of a triangular shape. And the way that your body recognizes them, in order to destroy them, hey, this isn't a human cell, it's a bad cell, it's a bacteria cell, is those antibodies are built to fit the exact shape, the triangle, And now your body can recognize, hey, this is a foreign invader. Let's go kill it. Let's go save the body. So just an example of why shape is so important. Antibodies, and for that matter antigens, are both proteins. So shape is very important to the function of a protein. Again, quick concept check, quick review of what we covered in this lesson. A list of several key terms to make sure you are familiar with them. And again, some concepts that, again, take a look at them, see if you can answer some of these questions, and if not, might be helpful for you to kind of go back and watch that video again, that part of the video, or to take a look at the PowerPoint and identify what this question is asking for. All right, thanks for tuning in. Have a good weekend.